So we've talked a little bit about receptor-mediated endocytosis, and I wanted to talk just a little bit more about phagocytosis and endocytosis, and also like maybe penocytosis and organizing the vesicles that are coming in, and how do we target them to the lysosome. So the difference between endocytosis and phagocytosis is the mechanics of how it happens. So in phagocytosis, we actually have an actin skeleton rearrangement that extends pseudopods around whatever you're trying to phagocytize. So very often there are cells that specialize in this. Cells such as like macrophages, one of the white blood cells that protects you from invading bacteria, or a dendritic cell. And so these cells will extend pseudopods around the thing that they're going to engulf. Think of this as like how an amoeba moves and eats, because this is left over from amoeboid movement, from the very beginning of cellularity. So some of the very first eukaryotic cells were amoebas, and this is how they move and eat. And some of our cells still now move and eat this way, like macrophages. And so there's some kind of food particle or some kind of cell, and the macrophage extends pseudopods around it by actin polymerization, so making their actin cytoskeleton longer on these finger-like projections, until they completely engulf the cell that they're going to eat. And once that cell is completely engulfed, now you've got it inside of a vacuole. And that vacuole can now go and bind with the lysosome, which, remember, it has a bunch of hydrolytic enzymes. And so it will start breaking down that bacteria. And you can go on YouTube and find a video, just Google macrophage eats bacteria, and you can watch them extend these pseudopods around this bacteria, and the bacteria is just sitting there inside of the vesicle for a short period of time, and then you see it start like really moving around and just like trying to get out. And the reason for that is because once that food vacuole binds to the lysosome, then all of these hydrolytic enzymes and acidic pH start breaking down the cell. It's going to really affect that bacterial cell. So phagocytosis is one of the ways that we deliver things to the lysosome. Another is one we already talked about, which is receptor-mediated endocytosis. One you probably haven't already heard about is something called autophagy. So if we break down the word autophagy, auto means self and phagy means eat. So this literally means eating yourself. So we can break down phagocytosis too. Phago means eat and cyto means cell. So this is cell eating. Cell eating, and this would be eating yourself. And so eating yourself doesn't refer to the entire cell. It just refers to maybe some organelle that has gone through its entire life and it just can't do its job anymore. Maybe something like a mitochondria that is not working anymore, or a peroxisome that has lived its entire life and now needs to be cleared from the cell. And what you can do is you can surround that with a vesicle, and and deliver it to the lysosome. And once it gets into the lysosome, of course, it's going to be digested. And that's a good thing for the cell, because you cleared out this non-functional mitochondria, but also the mitochondria is made up of fats and proteins and sugars and nucleic acids, and so you're going to have a bunch more monomers to make your polymers out of. So catabolism very often leads to the building blocks for anabolism. And so autophagy is something that many people haven't heard of before, but here we've got a picture of a autophagosome, and inside of the autophagosome on the left is a mitochondria, and on the right is a peroxisome, and both of them are inside of this autophagosome, and so they have been targeted for destruction by a lysosome. And then we did already talk about receptor-mediated endocytosis, clathrin-coated pits, and Rab proteins directing the uncoated food vacuole down to the lysosome through an endosome. So we went from clathrin-coated pit to a coated vesicle, decoding it, returning the receptors back to the cell surface, and then the endosome joining with the lysosome for digestion. And all of these processes, whether it is budding off of the ER, budding off of the Golgi, movement from the ER to Golgi, through the Golgi, endocytosed vesicles, receptor-mediated endocytosed vesicles, phagocytized vesicles, all of these things are going to depend on these tags. So we've got receptors for, so we talked about mannose-6-phosphate. So the mannose-6-phosphate has a receptor, and that receptor on the inside binds to the mannose-6-phosphate, and on the outside binds to an adaptin protein, and then through these Rab adapter proteins gets targeted to the correct vesicle, the correct final location, no matter if that's protein production and delivery to the proper portion of the endomembrane system, delivery to the cell surface and secretion of the protein out of the cell, or whether it's going the other direction through receptor-mediated endocytosis, phagocytosis, penocytosis, autophagy. All of this movement is going to be directed by these receptors, and which bind to the cargo on the inside, so they know what they're holding, and the adapters on the outside, so they know where they're going. So that's the endomembrane system and trafficking, and this is the meat and potatoes of cell biology. So I'm glad you guys stuck it out to the end. I know that 5 videos seemed like a lot for chapter 15. It was a meaty chapter, and I wanted to make sure that you guys really got a good taste of all the stuff that's covered in the chapter, and that's why we had to go with so much video. Thanks, and you can move on to chapter 16 if you'd like.